Hello friends, in our new video we continue to our lessons with kinetosome. It looks like centriole in structure. Have you recalled the structure of centriole? We said it made up of 9 triple groups of microtubules make a circle. It is structured at the base of the cilium and flagellum in cells which contain cilium and flagella. Means any kind of cell have cilia or flagella. Their base contain a kinetosome. The kinetosome has a vital role in movement of cilium and flagella. It also called as basal body. Why it called basal body? There's a question like this, because it located at the base of cilia and flagella. Structure of kinetosome in exam, it asked you, not right, it look like centriole. It made up of what? 9 triple groups of microtubules which are arranged around a circle, and function help movement of cilia in flagella in any cell which have this cilia and flagella, and their location is located at the base of cilia or flagella. Kinetosome. Vacuoles, they are membranous sac that exist in cell cytoplasm and vacuoles in some protists, whereby it is represented by contractile vacuoles. Contractile vacuoles, they can contract and relax. As they relax, they absorb water from the cytoplasm of organisms which live in fresh water like paramecium or amoeba, but as they contract, pump out this water, which drain the cells from surplus water and dissolved fecal material like amoeba and paramecium. It represents urination in animals. For example, give out excess amount of water out of the cell, but not pure water. With this water also discharge some dissolved waste materials. Which one? Contractile vacuole. But main function of this organelle is to provide osmotic balance in cytoplasm and environment. There are also temporary food vacuoles. Temporary food vacuoles formed when food is surrounded by a membrane of living organism. We can remember this function from nutrition in amoeba. If there's pieces of food, amoeba form pseudopodia, surround the food, take it into the cytoplasm, and as digestion completed in cytoplasm of cell, the vacuole disappear. Means the vacuole formed when food entered the cytoplasm. After food being digested, useful parts absorbed, non-useful part again discharged from the cell through a hole, and we will call it exocytosis in next page. And after vacuole disappear, that's why they call it temporary food vacuole. But temporary food vacuole mainly for animal cells, not for plant cell. Food is digested inside this vacuole, secreting enzyme from lysosome. As for plant cell, now the vacuoles are clearer than those in animal cell. It's a difference between plant cell and animal cell. In animal cell, vacuole size is smaller but number more. In plant cell, number less but size is bigger. They are small in young cells, plant cell too, wide in adult cells. They contain juices of different dissolved materials known as cell sap, and means materials which cell not need them at that time. They store it in vacuoles, so we call them storage vacuole. Mainly storage vacuole for plant cell, temporary food vacuole for some unicellular organisms like amoeba or animal cells, contractile vacuole again for amoeba or freshwater organisms. three kinds of vacuoles according to their function and maybe location. Hi friend, by vacuoles we completed our living contents of cytoplasm, organelles. What we need to focus on, their location, structure and function. Sometimes we need comparison between different organelles. Now, non-living content of cell, they are temporary contents, temporary vacuole. For a limited time they are found in cytoplasm, called cytoplasmic deposits, materials which are stored in cytoplasm of cell for a limited time. We call them cytoplasmic deposits. They are mainly formed by metabolite materials of pilot deposits. Like what? The organelles we studied on, they all living content of cytoplasm, but what they produce, non-living contents of cell. Ribosome living, but proteins produced by ribosome non-living. Chloroplast living organelle, but sugar which produced by chloroplast, or starch formed in leucoplast non-living. Or enzymes secreted by Golgi, complex carbohydrates secreted by Golgi, or cellulose and other components of cell wall which are secreted or formed in dictyosome. They are all non-living materials of cell. And means materials which produced by metabolic activities of these organelles. So there are 5 kinds we want to focus on. Lipid drops in fatty or adipose tissue in liver cells or adipose tissue. Lipid excess amount of fat in animal body stored in fat tissue, we know from fatty people in their abdomen, or liver cells. And as we eat a lot of food continuously, first converted into lipid, first time stored in liver, but when liver is not enough to store all of this food material, after stored around the abdomen and so on. Second, formation represented by glycogen, as in liver cell. Glycogen also, it's a carbohydrate, but complex carbohydrates like starch. Starch, storage form of glucose for plant cell, but glycogen, storage form of glucose in animal cell. Also glycogen formed when there is excess amount of sugar in animal body and stored in liver cells. Number 3, proteins stored in glandular cells, gland which their function secretion in the form of secretory granules. These granules are periodically released outside the cell. A glandular contains the release hormones in 2 forms, in fat, we call them steroid hormones, you may remember from smooth endoplasmic reticulum, or in protein nature. So glandular cells, we know gland means secretion, so they secrete these stored proteins in form of secretory granules. Periodically means not continuously, else body need them. Deposits of pigments, we remember pigments, colorful substance and coloring. Some cells produce pigments, as in the skin cells. Skin cells, there are melanocyte. Skin cells, they contain melanin pigment. So more pigment, dark color of skin, less pigment, less darkness in skin of organisms. Also the color depends on what amount of pigment which is stored in these cells. Enzymes, hormones, and some vitamins are also cytoplasmic deposits, which are granular, spherical, or oval in different shape. And these are materials are surrounded by membranes, as in the case of neurosecretory granules. We know neurons, they continuously secrete some materials out of cell, mainly hormones. or some other body part secrete enzymes, or like liver producing some kind of vitamins. Nucleus, you remember we said there are three main parts of eukaryotic cells: cell wall and plasma membrane, and after cytoplasm. We completed cytoplasm, living and non-living contents. And last part of eukaryotic cell is nucleus is the most important cell component in living organisms. It's essential for life. Why? Because cell survival depends on exchange between nucleus and cytoplasm. Material exchange between nucleus cytoplasm stop, so cell die. The cell without nucleus lives for a short time, then degenerates like adult red blood cells. Because when we compare red blood cells to white blood cells, red blood cells have shorter lifetime because they lost their nucleus when they become mature. Cell nuclei can be different in shapes. This different in shape depend on cell shape. We remember from before we said cell shape also depend on its function. It might be spherical or oval or lobed or irregular like white blood cells. Lobed we can see in the shape a cell with nucleus and that shape, or sometimes maybe not only two lobes, maybe three lobes also, some kind of white blood cells, we will see them. Irregular in shape, some cell we're going to study in connective tissue, their nucleus in that form, or like ovoid like an egg, or can be spherical. The nucleus is the largest distinctive organelle inside the cell. We may remember after discovery of cell, the first structure discovered in cytoplasm is nucleus, which is discovered and described by Robert Brown. Size differs according to the cells. Its size depends on size of cytoplasm, means bigger cell, more cytoplasm, and bigger nucleus. Most cells are mononuclear cells, and mainly one cell has only one nucleus. In some cases, the cell can be dinuclear, means has more than one nucleus, like cartilage cell, liver cell, and neuron cells. My friend, you can tell also there are skeletal muscle cells with more than one nucleus. Can't we give example like this in ministry exam? No. So in ministry exam, there's kind of question. Write an example or two examples for the followings. So if ask you to write example for cells with more than one nucleus, you have to give one of these three examples, but not any other cell, you know. The nucleus is central in stem cell. It's a short information, but a chapter question with 2 marks. It has side or peripheral location in some secretory cells. Sides or peripheral means like in fat cells, because cytoplasm full of fat, there is a little space for nucleus. So nucleus located beneath the membrane. And this we call it peripheral. Peripheral means on the surface. Like fatty cells and mucous cells, the nucleus consists of following components. It's a thin double-layer membrane. Nucleus means it covered by 2 membranes, as in mitochondria and in chloroplast. It surrounds the nucleus and it has its own physical and chemical properties. It regulates exchange of materials between nucleus and cytoplasm through micropores that allow passage of particles. Function similar to cytoplasm, but for cytoplasm we said exchange of material between cytoplasm and external environment. For nuclear membrane, between cytoplasm and nucleus. This membrane has selective permeability. You may remember we studied on 4 types of membranes. So when asked about to give example for this kind of membranes, selectively permeable, example for nucleus. Selective permeability means allows passage of some materials but not others. This membrane surrounds the nucleus in all cells except for bacteria and blue-green algae, prokaryotic. We know prokaryotic cell genetic material not covered by membrane, so they have no nucleus, but they have nucleoid. It has no nucleus, but it has nuclear matter just. Nucleoplasm, as we draw nuclear membrane. Nucleoplasm, you may remember we said cell membrane filled or cavity of cell filled by cytoplasm, cavity of mitochondria filled by matrix, cavity of chloroplast filled by stroma, while nucleus filled by a colorless gelatinous liquid, we call it nucleoplasm. Which other nuclear contents are distributed? What are these nuclear contents? They are nucleolus and chromatin network. So the nucleus has one or more nucleolus. If we think about this plasma membrane, in center a big nuclear membrane filled by nucleoplasm, but this nucleoplasm filled by what? Chromatin network, a filamentous irregular material fills the nucleus. We call it chromatin network. The nucleus has one or more nucleolus. For example, onion cell nucleus has 4 nucleoli. The nucleolus is a relatively big spherical structure inside the nucleus. Nucleus inside the cell, nucleolus inside the nucleus. This information onion has 4 nucleoli. Chapter questions, my friend. Nucleus, relatively big spherical structure in the nucleus. It consists of protein and RNA. Genetic material free, but in some points, in addition to chromatin network, there are some other proteins. In addition to proteins, some more RNA, and it looks like dark and spherical structure, and we call it nucleolus. So this dark and spherical structure in nucleus, we call them what? Nucleolus. What it contains? It contains chromatin network plus protein plus RNA. RNA, a nucleic acid, like assistant of genetic material, DNA. And this long filaments of chromatin network, in real it's DNA. But in nucleolus, in addition to DNA, there's RNA and some other proteins. So like dark spherical structure, and we call them nucleolus, and there are 4 of this nucleoli in onion cells. The number may change from one cell to another. Nucleolus has vital role for ribosome, which is responsible for protein formation. We maybe know ribosome also consists of RNA plus protein. Also in nucleus, DNA, protein, RNA. Chromatin network is an interconnected and irregular filamentous structure. Filaments of chromatin network appear during cell division, forming a specific number of bacillary structure known as what? chromosomes. Simply we need to know the shape is chromosome. They carry genes, a specific part of chromosome which carry one specific property like eye color or flower color or shape of color or position of flower on plant's body. Each of these genetical features controlled by one gene located on chromosome and transport genetic features from one generation to another. Therefore, chromosomes are very important because of vital role in genetics. We will study on these genes and chromosomes in chapter 5 details, or reproduction, development, and mutation or maturation. Mutation also in genetics we will study. Chromosomes can be seen only during cell division, and if there is no division, we see genetic material in that form, but as division starts, they change into basilar structure or in that shape. The number of chromosomes differs in living things. Living beings have fixed number of chromosomes, and one property which make organism in different species is their number of chromosomes. In their somatic cells, there are two types of cell in organisms: somatic cell or gametes, or we can call them reproductive cells. So living beings have fixed number of chromosomes in their somatic cell, in blood cell, muscle cell, bone cell, skin cell, eye cell. They all contain same number of chromosomes. Number of chromosomes change from one organism to another, not from one cell to another. So a kind of worm with two number of chromosomes is the least number of chromosomes among organisms. The number of chromosomes in Spanish butterfly 380. 12 number of chromosomes in homefly, 26 in frog, 80 in pigeon, 64 in a horse, and 46 for human. This number not to memorize, but we need to know. Maybe we need number of chromosomes just in human. It must be noted that this number of chromosomes represent the somatic cells. Number of chromosomes in gametes or reproductive cell is half. What does it mean? For example, human egg has 23 chromosomes, while somatic cell have 46 chromosomes. Means in a muscle cell there are 46, but reproductive cell and egg cell contain half number. Why? It formed by or a different kind of division, which we're going to study after. But in chapter question about these numbers, that only question about Spanish butterfly, it asks about if there are 380 chromosomes in a somatic cell of Spanish butterfly, what is the number of chromosomes in egg cell? We just divide it by 2, and 190 chromosomes for an egg cell in Spanish butterfly in a chapter question. And we need to know from that part, somatic cell, muscle cell, bone cell, blood cell has full number, but only reproductive cell has half number of chromosomes. It results from fusion of egg cell with sperm cells. The chromosome have fixed size and shape in living organism. Its length ranges from 0.2 to 50 micrometer. Means what? One egg cell, one sperm cell, they are both of them gamete with half number and half number, but new organism formed by fusion of these two numbers to form a 46 number of chromosome again. But new cells, again, they divide into two to form again gametes 23 and 23. And this 23 fuse with another 23 again to produce 46 chromosome number. Again, in that way, life continue. Comparison between animal and plant cells. Animal and plant cells differ in some areas. You may remember when we study on organelles, cell wall and different organelles, we talk about if they are found just in plant cell or animal cells. Show the basic area similarity or differences between animal and plant cell from a structural point of view. This comparison is important, and it's one of the most frequent asked questions in ministry exam. Comparison between plant cell and animal cell. Cell wall, in addition to a thin plasma membrane, also plants has what? A thick cellulose wall consisting of lignin. We remember cell was made up of cellulose, and after taking it with lignin to form secondary wall, but in animal cell there's only thin plasma membrane. Plastids, chloroplasts, which give associated or responsible for photosynthesis, leucoplasts, which convert glucose into different organic materials, and colored plastids, chromoplasts, which give color to fruits and flowers, found just in plastid, but in animal cell there are no plastids. Centrioles or centrosome, no centrioles in plant cells, but centrioles exist in many animal cells. They have a role in cell division, in movement of chromosomes. Vacuoles are few in plant cells but large in size. They occupy the whole size of adult cells. Most of cytoplasm occupied by vacuoles in adult cells. There are many vacuoles in animal cells but small in size, spread in cytoplasm. Cell division, we will study this detail in end of chapter in cell division, but simply during cell division the cellular plate is created. When we study on cell wall, you may remember we said first in cell division nucleus divide, after division of nucleus completed, division of cytoplasm start, but in plant cell it starts from cytoplasm in to outward until it reaches the cell wall and divides cell into two. But in plant cell, a spherical plant cell, from two sides, like a pressure happen on cell or pulled by some inner materials, it moves inward like pinching of squeezing a balloon until cell divide into two and two cells obtain it in plant cell, means in a spherical form, after change into that form and completely divide into two. A difference from the center outward, but in animal cell cytoplasm bend from outside and moves inside. A difference about division of cytoplasm in plant cell and